We expect that you are already very familiar with the information presented in this image of DNA. There are 3 facts about DNA that we'd like to highlight here. First, the building blocks or subunits of DNA and RNA are nucleotides. A nucleotide is made up of a sugar, deoxyribose in DNA and ribose in RNA, a phosphate group, and a nitrogen-containing base, adenine, guanine, cytosine, or thymine, which is replaced by uracil in RNA. Second, phosphate molecules have a negative charge, and the phosphate groups in DNA make it a negatively charged molecule. Because DNA is a charged molecule, different sized fragments of DNA can be separated using gel electrophoresis. The importance of this will become clearer as we move through this course. And third, the DNA molecule is oriented such that each of the 2 strands has a 3' end and a 5' end. These designations refer to the numbering system for the 5 carbon atoms in the sugar molecule. The 2 strands in the DNA double helix are antiparallel. That is, the 3' end of one strand is opposite the 5' end of the other strand, and vice versa. This is what is meant by the term antiparallel. You will remember from your study of DNA replication that in replication the newly synthesized DNA strand extends from 5' to 3'. In humans, there are 22 pairs of autosomes and 1 pair of sex chromosomes. Pictures of chromosomes seen through a microscope often have a banding pattern that is the result of staining used for chromosome identification. Looking at one chromosome shows that the bands are used to identify chromosomal regions. The short arm of the chromosome is referred to as the P arm. A Frenchman devised this naming system, and P stands for petite or short. The long arm is the Q arm. Focusing on a small segment of chromosome 16 allows us to talk about the DNA structure of the genome. Genes are located along the chromosome, but chromosomes contain a lot of DNA that is in between the genes. This is called intergenic DNA. Looking more closely within a gene, there are both exons and introns. DNA within a gene is transcribed into RNA, but not all of that RNA is translated into protein. The intronic RNA is spliced out, and the exonic messenger RNA leaves the nucleus, goes to the ribosomes in the cytoplasm, and is translated into protein. This cartoon of a gene is just a cartoon, and if we could look more closely into what the DNA looks like, we would see the sequence of A's, C's, T's, and G's that make up the code of DNA.